Hello, everyone. I am Shubhra Malik, and I will talk about my research on targeting microRNAs for cancer therapy. Now, first, let's look back in time and see how the field of gene therapy started. It was in 1953 when Rosalind Franklin imaged the crystal structure of DNA, and later James Watson and Francis Crick used this information to propose that DNA has a double helical structure where two strands bind to each other by complementary base pairing and arrange in a right-handed helix. In 1968, Francis Crick stated the central dogma of molecular biology, which explains the flow of genetic information. And here, the genetic information stored in the DNA is first transcribed into RNA. Now, this RNA moves to the cytoplasm where it undergoes translation to form the proteins, which are the functional units for all biological processes. And when we talk about RNAs, there are two types of RNA, that is coding RNA and non-coding RNAs. Now, coding RNAs, they move to the cytoplasm and are translated into proteins, but they constitute only 2% of our genome. So what about the rest of the 98%? Until recently, that 98% of our genome was called as the junk DNA. And in the last decade, about 50,000 non-coding RNAs have been discovered. And now we know that these non-coding RNAs, they are not junk and they are essential to regulate several physiological processes. Now, there are several non-coding RNAs, including microRNAs, long non-coding RNA, circular RNAs, transfer and ribosomal RNAs, which are present in the cytoplasm. And today, I'm going to specifically focus upon microRNAs. Now, these microRNAs are small non-coding RNAs, which are about 20 to 25 nucleotide length. They themselves do not form any proteins, but they regulate the protein formation of other coding messenger RNAs. Here, microRNAs, they associate themselves with certain proteins to form an RNA-induced silencing complex or RISC assembly. And the microRNA will guide this RISC assembly to the target messenger RNA, where it will bind to the messenger RNA and then induce its degradation. Interestingly, one microRNA can inhibit the translation of several different mRNAs. Now, why are we interested in these microRNAs? Here, the normal levels of microRNAs are essential for fundamental biological processes such as growth and development. But in case of cancer, where the cells are rapidly growing, the levels of some microRNAs are dysregulated. And when we say dysregulated, it means they can be either upregulated or downregulated. And here, microRNAs which are upregulated in cancer, they are known as oncomeres. And in order to correct the level of these dysregulated microRNAs, we can use either antimeres, which will bind to the highly expressed microRNAs and inhibit them. Or in cases where the level of microRNAs is low, we can administer the mimics in order to restore the normal functioning. Now, both these strategies have opened up a new field called microRNA therapeutics. And this is the list of antimeres as well as the microRNA mimics, which are currently in clinical trials for treatment of cancer like lymphoma, lung cancer, breast cancer, and many more. And my research focuses upon designing new antimeres in order to inhibit the oncomeres, which are highly upregulated in cancer. And I work on designing peptide nucleic acids, which can bind to the target microRNAs by complementary base pairing rules. Now, these PNAs were discovered in 1991 by Dr. Peter Nelson as mimics of DNA. Now here, DNA or RNAs have phosphodiester backbones, which makes them negatively charged. But PNAs are neutral as they do not have any negative charge in the backbone. And we can design these PNAs to target any RNA in a sequence-specific manner. and these PNAs will bind to the microRNA and will inhibit its function. And one of my projects is specifically on targeting microRNA 155, which is highly upregulated in lymphoma and leukemia. And here, this is the sequence of oncomere 155, which is about 23 mer long. And here I designed a series of PNAs with different modifications in order to efficiently target this microRNA 155. Now, we make these PNAs in our lab using solid phase synthesis, then we purify them using chromatography, and finally confirm their quality by checking their molecular mass. This slide provides a bit more detail on how we make these PNAs. And based on the sequence, we add each unit step by step on a resin, which acts as a solid support. And in order to ensure that the reaction was complete at each step, we use a color-based test called Kaiser test. Now, if it's yellow, that means we can go ahead and add the next unit. But if it's blue, that means that the reaction was not complete, and then we need to repeat the same step again. And once we have added all the units in order to obtain the complete sequence, we can cleave the PNA from the resin and then collect it for further purification and testing purposes. Now, if we directly inject the PNAs in body, it will be rapidly eliminated within 15 minutes. Now, in order to prevent that, we encapsulate these PNAs in nanoparticles, which also helps in delivering the PNAs to the tumor site to inhibit the cancer-causing oncomeres like microRNA 155. And here we use PLGA polymer to make nanoparticles, which is approved by US FDA. And we make these nanoparticles using the double emulsion solvent evaporation technique. And here we add the PNA, which is in water, into the PLGA polymer, which is dissolved in an organic phase. And then we sonicate it to form the emulsion. And then we add this emulsion again into the water to form water and oil in water double emulsion. And this results in the formation of these oil globules containing the PLGA polymer and PNA together. And finally, we collect these nanoparticles containing PNAs inside them, and then we can use them for further testing. Now, this is how these PNA containing nanoparticles look like under a microscope. Their size is about 150 nanometers, which is small enough to accumulate in the target tumor sites. And here we also determine the amount of PNA present in these nanoparticles by loading analysis. And then we study that how these PNAs are released from the nanoparticles over time. And here you can see that the nanoparticles start to release the PNA within 15 minutes and then releases the entire amount of PNA encapsulated in them by about 24 hours. Now, we test the activity of these PNA-containing nanoparticles in different cancer cells and then perform several different analyses to select the best reagent. And once we have our best reagent, the final goal is to test their ability to inhibit the tumor growth in vivo. And for this, we first developed these mice bearing the tumors by injecting the cancer cells subcutaneously. And after 4 to 5 weeks, we can see a tumor, a solid tumor on the site where we had injected the tumor cells. And now we inject our PNA-containing nanoparticles into the mice systemically and then monitor the growth of the tumor over time. And in comparison to the untreated tumors over here, the PNA NP treated tumors show reduced and slower growth. So overall, we are designing a new strategy to treat various cancers. Now, there are several microRNAs which are upregulated in cancer, and we can design a PNA to target any microRNA which is upregulated. Also, this represents a novel personalized approach for treating cancer, which can be extremely beneficial for the patients who do not respond to other anti-cancer therapies. That will be all. Thank you guys for listening, and please let me know if you have any questions. I'll be very happy to answer.